Hi, my name is Zachary Toomey, and this is my YouTube project for ENTO 320 titled Ultraviolet-sensitive opsin proteins enable Apis mellifera to effectively locate food. An introduction to Apis mellifera, belong to the order Hymenoptera and the family Apidae. They have a common name of Western honeybee. They originate from Asia, across the Middle East and Northern Africa, but they're not native to the Americas. They were actually introduced to the Americas through humans, not naturally. When it comes to their size, they are about middle of the pack. They are smaller than the giant honeybees, the subgenus Megapis, and they are larger than the subgenus Micropis, which is the microhoneybees. They are also larger than their close relatives, the Apis cerana. Apis mellifera are very important due to multiple reasons. Two main ones being their ability to pollinate plants and their ability to produce honey. That source goes a little bit deeper into the ability of the impact of their pollination. Apis mellifera also have trichromatic vision, which means they can see in three different colors. They see ultraviolet, blue, and green. This is due to opsin genes. Opsin genes are photoreceptor proteins that are found in cone cells in their eyes, responsible for their colored vision. That article goes a bit more in depth into the purpose of opsin genes and how they work. Thanks to these opsin genes, they're very important to see nectar guides. Nectar guides are markings on flower that indicate nectar being present in the flower and allow the bees to make decisions, as we'll discuss later in this presentation, about caloric rewards through these nectar guides. My research question for this project is, what effects do ultraviolet-sensitive opsin proteins have on honeybees, Apis mellifera, ability to find food? My first content slide comes from a journal from Townsend and multiple other authors. This article was published in 1998 and expressed the opsin genes to characterize the spectrum at which they're able to see these colors. They express these genes in different flies, the Drosophila, I probably butchered that pronunciation, genus of fruit fly. And this article proves that supports the ability for these Apis mellifera to see at the ultraviolet wavelength. As seen in the figure, the Apis UV wavelength is around 360 to 370 nanometers and very sensitive to that wavelength. My second content slide is from an article from Ray and Vasmosi. Also apologize for the mispronunciation of that name. This figure shows a relationship between different UV reflectance of these nectar guides that we discussed earlier on the flowers of pollinators, specifically Apis mellifera. This study focused on Apis mellifera and a specific species of flower. And they manipulated the UV reflectance of the flower through different treatments and found different visitation rates per treatment. This supports the fact, or the idea, that the different UV reflectance correlates to pollinator visitation from Apis mellifera. This content slide shows that Apis mellifera visitation to flower types can vary based off their caloric reward. This study took three different sections, three different flowers with different nectar guides with different caloric rewards. In section 1, the caloric reward was the same, and it found that the visitation from honeybees was very similar, 300 to 320 visits to the two different flowers. Section 2 had a slight caloric advantage to the blue center flower. And then in section 3, there was a slight advantage to the white center flower. And what they found was that the Apis mellifera honeybees were actually able to remember the caloric reward based off of the UV reflectance of the flower, and remember that that specific flower was not very rewarding, and were more likely to visit the more caloric rewarding flower to make the most to get the most caloric value out of each visitation to the flower. My conclusion is that the opsin proteins that have been characterized in Apis mellifera genes to be sensitive to ultraviolet wavelengths are used by these honeybees to find and visit flowers, and based on the visitation rewards, caloric decisions can be made. These proteins allow for honeybees to locate and utilize food optimally. This was seen through my first resource showing that the Apis mellifera were sensitive to the UV wavelengths. And then through the second one, that they use these UV wavelengths to find and visit flowers. And through the third one, that they can remember these flowers based off the UV reflectance and make caloric decisions to utilize food, or to locate and utilize the food optimally. The significance is that Apis mellifera can use this to find food optimally, which will, and I can theorize that this will increase their life expectancy thanks to these opsin proteins, and that this is very important to understand the feeding behavior of honeybees when studying them. And here are my 5 peer-reviewed sources found throughout my presentation. Thank you so much.